Welcome to the course on wastewater characteristics. In this lecture, I will deal with the sampling examples, and I will share with you two very nice sampling examples, one from the full scale and one from the laboratory scale. The first example is about sampling campaign that we performed to obtain new data to support the modeling study of the city of Poreč in Croatia. The scope of sampling included sewerage systems, 4 catchments, and the 4 wastewater treatment plants, and the stretch of the coastal sea of 23 kilometers. This is an image that depicts the area of sampling, the area where 4 wastewater treatment plants were built and where the sewer system was reconstructed and extended by the length of 217 kilometers. On the picture below, you can see with the blue squares marked many of the beaches which are having a blue flag for safe bathing. The sewer systems included 4 catchments, each having one wastewater treatment plant. And on this slide, you can see these 4 catchments in different colors. And in the red areas, you can find out where are the current discharges of wastewater and wastewater treatment plants. In this project, 4 wastewater treatment plants were built of the total capacity of 145,000 person equivalent, and all implemented similar technology based on membrane filtration. In the part 1 of this case, I will share with you information on sewage and wastewater treatment plant sampling. In this study, we have performed 3 sampling campaigns. 2 without wastewater treatment plants in place, so before the wastewater treatment plants were built, and 1 after the treatment plants were built, but during their startup. The first sampling started in January 2019, which covers winter conditions, 7 days at 4 locations at seawater discharges, and in total we collected 336 samples. The same protocol was applied in July 2019 during the summer period, again with the 7 days, 4 locations, and 336 samples. And in January 2022, which means again in the winter, we performed sampling analysis, sampling program at 4 different locations of wastewater treatment plants, and each campaign was 1 day, so 1 day at each plant, which was in total 4 days. Influent and effluent we sampled, and we collected 96 samples. At each point, we collected information on 15 parameters, which means in total more than 10,000 wastewater quality analyses and more than 400 analyses of seawater quality. The parameters that we decided to examine in this study were COD, COD filtered, BOD and BOD filtered, total suspended solids, inorganic suspended solids and organic suspended solids, total nitrogen, ammonia, nitrate, nitrite, total phosphorus, phosphate, pH, and electroconductivity. We created a sampling schedule. This sampling schedule was mostly determined by the number of samples, by the budget available, and by the time available for the execution of the study. So this is an example from one of the 4 wastewater treatment plants in Versar. In the winter 2019, the sampling program was 7 days duration. We collected 12 samples per day. These samples were 2-hour, 5-minute composite samples. 2-hour composite sample means that we could have 12 samples per day, and each of these samples is collected each 5 minutes, so we needed 12 samples, small samples per hour, which means 24 small samples per 2 hours. So for 2-hour composite sample had 24 small samples in it. And we collected 24-hour composite samples of the entire day. And there we took analysis of BOD5 for all 4 wastewater treatment plants. And on this slide, you can see an example of the schedule that is containing the location of samples, the time of samples, the frequency, and also the amount of samples that are collected. On each 4 locations, we installed automatic samplers for collection of composite samples in winter and summer 2019 at location of 4 wastewater treatment plants. And you can see the locations and sampling devices at each of these locations on this slide. In the winter 2002, that was the 3rd sampling campaign, remember, and this time wastewater treatment plants were already built and just started up. So at the wastewater treatment plant, there are already automatic samplers at the point of influent and at the point of effluent. So we use the existing new automatic samplers to collect samples from the influent and effluent of each of 4 wastewater treatment plants. After we collected all these samples, we immediately preserved them and transported them to the lab where they were analyzed. And they were analyzed in 3 different labs. In the winter 2019, they were analyzed locally at the Institute of Agriculture and Tourism in the city of Poreč. In the summer 2019, we took samples and transported them to the faculty at University of Zagreb and analyzed the samples in their lab. And in winter 2022, wastewater treatment plants were already built, and at one of them there is a very nice laboratory, so we analyzed all samples at the laboratory of the wastewater treatment plant. Part 2 deals with the seawater sampling. For seawater sampling, we used this beautiful boat, which is a property of the local Institute for Oceanographic Research, which were our partners in the project. And the boat is equipped with all kinds of analytical equipment and also analytical devices, but also sampling devices. In this particular case, we were really interested in indicators of fecal pollution, and such samples were collected by this special sampling device that allow us to take samples at a different depth. Samples were immediately preserved and transported to the laboratory of the institute and analyzed for Escherichia coli and coliforms and intestinal enterococci. These are just some images of the results, and sampling locations were determined in a way that are the same sampling locations in the winter and summer 2019 and winter 2022 campaigns. There were 4 sampling locations. These 4 sampling locations were identical at all 4 wastewater treatment plants, and they were taken at the point where the long offshore discharge of the effluent from wastewater treatment plant is located. The point A is offshore discharge location just above the mouth of the pipe which discharges effluent from wastewater treatment plant. Points B and D are points 150 meters in direction of currents, upstream and downstream of currents. And the most important is actually point C, which is a point 300 meters from the coastline, which is by the law determined distance from the beach at which water quality should be of a certain quality prescribed by law. Based on that, we developed a sampling schedule for all 3 campaigns. As I said, it was 1 day at 4 offshore locations, 12 samples per day. We collected 2 hours, 5-minute composite samples and 24-hour composite samples for analysis of BOD. In this example, I presented you with the sampling procedures in which we sampled a sewer system, wastewater treatment plants, and coastal sea. The results of these sampling procedures is not falling under the scope of this particular course, but you can find it in a course on modeling, where you have a case study from Poreč. And there we showed also the results of this sampling procedure. So if you are interested, please go to the modeling course and you will find all the results from this sampling program. And now I would like to move to the second example. The second example is about a lab scale experiment that we have done at IG Delft Institute for Water Education as a part of our PhD and Master of Science programs. The objective in this particular example was to obtain the data on process conversion rates regarding enhanced biological phosphorus removal during the anaerobic-aerobic batch activity test. And in this case, our scope was actually the reactor, 2-liter batch reactor, which was used to enrich phosphorus accumulating organisms. In this study, we developed an experimental sampling form which really contained all necessary information to perform the test, but also for any other person or students to repeat the test using exactly the procedure which is prescribed in this form. The part of the form is sampling schedule. This is a sampling schedule as an example of anaerobic-aerobic EBPR batch activity test, which is in total duration of 270 minutes and has sampling intervals between 5 and 45 minutes. This is very interesting to study such an experimental procedure because sometimes, and specifically at the beginning of different experimental phases, you need to take samples more often. And as the test continues, you don't need to take samples more often because concentrations changes and dynamics of the test is getting slower. So it's not necessary to take samples every 5 minutes. But for this, you need to have some experimental experience and to read lots of papers to find out what is the most optimum way to perform lab scale tests. The minimum duration between samples here was 5 minutes because we couldn't go less than 5 minutes because we need time to perform sampling and to store the samples and to handle samples in proper way before we take a next sample. So for very skillful technician or researcher, you need minimum 5 minutes to get ready for taking a new sample. And therefore, the 5-minute interval was the most frequent interval in this test. As I said, as the test continues, the need for more frequent sampling is less, so we can take samples at 10 or 15 or 30 minutes interval, at the end 45 minutes interval. In total, we took 21 SNAP samples for acetate and phosphate because these were the two most interesting parameters that we need to follow in this test. We also took 4 samples of ammonia, PHA, and the glycogen because these parameters are also interesting for the test, but PHA and glycogen are parameters that you don't need to sample every 5 minutes. You can sample it at the beginning and at the end of the test because in this particular case, we were not interested on dynamics of PHA and glycogen, just on the total consumption or production of these parameters during the test. And we took 3 samples in triplicates for mixed liquor suspended solids and mixed liquor volatile suspended solids. Why 3 samples? Because we take a sample at the beginning and at the end of each phase, so it's in total 3 samples. And the triplicates because normally when you analyze solids of the biomass, then you need to perform at least test in triplicates so that you have higher accuracy. In this particular scheme, you can see that we really organize everything that can be seen on one sheet, and there's lots of information on it. If you need more information on this, take a look into last course in this series on experimental methods in wastewater treatment, and you will find such sampling sheets to execute large number of different batch tests used for, in this case, activated sludge dynamics and activated sludge activity tests. On this photo, you can see the experimental and sampling equipment used for this particular case. On the left-hand side, you can see two reactors from which one is used for biomass. At this particular photo, you don't see the biomass in the system because the photo was taken just before we started implementing biomass in a reactor and then taking the samples. But that is not so important. It is important to see that you need to be very well organized. Before you start the test, you need to plan everything accordingly. You have, next to the reactor, you can see that piece of paper. That's this sampling experimental sheet that I just explained to you a minute ago. And on the right-hand side, you can see a trolley with all eprouvettes and vessels needed and containers to store the sample, to preserve the sample. There is also ice that you put the samples in so that you stop biological chemical processes taking place in the sample, and so on and so on. On this final slide of this presentation, you will find snapshots from our video materials presented in course number 18 in this series, Experimental Methods in Wastewater Treatment, and these snapshots are taken from the activated sludge activity tests. And you can see our analyst on the left-hand upper picture taking a sample with the syringe from the biomass in the reactor. The other two pictures with the red and blue circle is actually preparation of the sample, in which in a red circle sample is directly poured into a container without filtering and used for analysis which are needed to analyze the biomass. And in the other example, which is in the blue circle, you can see that analyst is using a filter and filtering the sample and only collecting the filtrate, which is free of the biomass and used for other type of analysis. In both cases, containers are placed in ice and stored and immediately after the test taken for the analysis, so to minimize the holding time. As I said, more of such examples you will find in course number 18, and I recommend you really to consult these procedures if you are doing any type of research involving experimental methods in wastewater treatment. Thank you for listening, and this is the end of presentation on sampling examples.